Hello, hi. And I said begin by covering my sexaholic portion from yesterday. All right, before that, how much of ATP was produced from my workout? I roughly had 10 minutes of step aerobics. And my heart rate went up to 24. And then I did high low after a drink of water, and it went up to 25. So approximately 165 calories I burned. Or at 165 calories divided by 0.012 calories per ATP molecule for per so how many ATP molecules are produced? So divided, so that's a lot of ATP molecules. So my textbook may not be all accurate. So I got 13750 ATP, which was used by my human body after being produced as cellular respiration energy. So it's in your best interests to consume, you know, glucose-rich fruits and that sort of thing. Of course, protein is essential because you've used up. I mean, it has to replace. And my last part was my sexaholic portion, and I had some me time, and sort of yada yada yada. I ended up having a good time. My husband was busy watching Zero Dark 30, and I had to resort to other means. What can I say? I mean, if the man's not around, then the woman has to make do for herself. And so that resulted in a nice end point. And my heart rate went up to 19 heartbeats for 10 seconds. Considering, I mean, really nice endpoint, and that explanation could be ATP production leads to, all right, signal transduction. We all know signal transduction. I've dealt with this before, have discussed it. So it ATP as an energy source, muscle function, sexual activity is a form of physical exertion that uses a significant form of energy which comes from ATP. All right, and of course, there are so signal transduction is a process where a cell receives and converts an external signal into a specific internal action. And in this case, well, yeah, it's the so ATP becomes CAMP in signal transduction. So the enzyme is adenyl cyclase. Yeah, adenyl cyclase, and so this is all a part of cellular respiration. But I've connected all this a little bit less, efficiently on my LinkedIn webpage. But there's more. Of course, there are neurotransmitters, glutamate, and glutamate is actually an amino acid, but neurotransmitters. It's also a neurotransmitter, I think, if I'm correct. Glutamate. Double check, please. Glutamate, and there's the other neurotransmitters. So ATP is connected to CAMP through a biochemical reaction where the enzyme adenyl cyclase converts ATP into CAMP. This process is fundamental in cellular signaling, as CAMP acts as a second messenger to relay signals from outside the cell to the inside. So that is, that results in, making it epithelial, epithelial vaginal cells, inner, outer, inner, the triangular, and all this, those are the cells and cellular respiration, etc., leading to a nice endpoint. All right, so that is part one from yesterday. Part two, birth defects. All right, I don't know actually, but I yes, you see these people. I mean, people with limbs, with children with limbs, and children with, but and children with crooked feet, crooked foot, one foot and the other one perfect, and that sort of thing. But really, if I were to actually tell somebody, I really don't know. But I've kind of been sort of cued about these things, and primary amongst them, is, and before that, do I have any other notes to discuss? All right. Now, hydrolases. Hydrolases are enzymes, of course, ases, and they catalyze group transfer reactions. So this is Watson's. The Crick and Watson were the gurus of DNA and DNA structure. So hydrolysis. So if you consume ethanol, so you see the hydroxyl group here, water. Water is primary to all cellular reactions. Although, but there's, as I found out from the internet, there's urine water and water which is used up. So, OH. So ethanol has a hydroxyl group. So there could be, ethanol being attached, the hydroxyl group from the ethanol, and here the C2H5 ethyl could be attached here. So these are group transfer reactions, which are those involving groups attached by high energy groups, and a high energy group is transformed to an appropriate acceptor molecule. It becomes attached to the acceptor by a high energy bond. A group transfer thus allows the transfer of, for example, how energy present in ATP is transferred to form GTP, one of the precursors used in RNA synthesis. You all know RNA, of course, plant basis, but there's transfer RNA, there's messenger RNA, and the third one, oops, I'm not getting it. Third one is transfer RNA, messenger RNA. I have to look. All right. So those are all key RNAs used in DNA transcription and DNA, not in replication, but they would all, send the wrong message out. So please do not drink during your pregnancy. All right, I wanted my child, so I kind of, and even if you want your child, sometimes you think you can get away with it, and some people do. Right? It's just the symptoms that the child possesses. All right, but sometimes it could be costly. And caffeine. Moving on to caffeine. Caffeine has a structure. You all know purines and pyrimidines. Caffeine. All right, caffeine. Oh, this is my bookmark. Caffeine. This is caffeine, and this is guanine. Again, a mimic. So guanine is attached, instead of guanine, the human body is attaching caffeine. So AT. So on the web, I looked up what could be the cause of birth defects, and there's also from child health, crooked feet and limps and everything. There's a website called healthychildren.org, and it discusses in detail. And so guanine, all right. So it discusses, what actually happens there in detail, the medical aspects. So what could be happening? Guanine could be being substituted by caffeine. And again, DNA is present in mitochondria, and in the plasma of the cell as well, but in the nucleus of the cell, and it could be an ATP is involved in. Again, ATP comes in, so growth and development. Glucose is essential, so ATP is involved in. So I'll go back to here. And water is used in growth and development and metabolism for children and fetuses. All right, so water hydrolase that I discussed. DNA is in mitochondria, which is the energy producing part of the cell. For guanine replaced by caffeine. Before that, I suggest ATP comes in here as well in DNA replication. And I'll discuss that and go to my last part, and that will be okay, DNA ligase. And this they found out from E. coli. It's too close to call for so it's too close for comfort with human people. With DNA ligases, they join, right? They join. So they would be joining AT and GC. So they use ATP as the co-substrate. So glucose, so it's so important, glucose consumption, and that is and for guanine replaced by caffeine, demethylation could be a possible. You'll have to see. CRISPR technology is wonderful. Even yesterday, the Globe and Mail had a huge center for CRISPR technology. But um, it's hard. It's too close for comfort. Again, you could kill the wrong genetic string. And so a swab test is good. So you can figure out the defective gene and hopefully introduce the correct, but there's no correct gene. That's what the internet says from the Human Genome Project for, mechanism of limbs. And they're all each one different part. So string of, a huge list of DNA genetic strings. So, um, um, so you cannot make it a dominant trait. You cannot hope to make this a recessive trait, and the dominant trait takes over, and the child does not have a pronounced limb, but a small limb, and goes around quite fine. But that is not a possibility. So demethylation accompanied by amination, but only to guanine, which mimics or also known as caffeine, which is mimicking the guanine, is hard. So I have hit upon de-ethylation is okay, all right, because alcohol coming out we could hope for just enzymes. So that's all dealkyl. That's all, list is all there. You can check with your health food store or the internet and see all of these factors. So delta G, Gibbs free energy, determines whether it's the AT. So purines with pyrimidines, right? AT or GC. So guanine is being mimicked by caffeine. So guanine is a double. The benzene-like structure with the 5 carbon atom structure, those two join together. So delta G can be based on glucose intake and related ATP production. And again, I looked into this, just before when I began, and the temperature would be the body temperature of the child, and delta G is equal to delta H minus T delta S, and delta H can be, if you're basing it on just glucose and adding enzymes to correct the guanine, which is actually caffeine, then delta H could be MCP delta T of glucose. Delta S in increase in disorder is increase in entropy. So delta S, would be, you could find out from experiments. So molecular, if you need to make a molecular transformation, then, but enzymes don't need. Again, this is activation energy, right? So delta S is connected to activation energy. So activation energy, I found out for enzymes, I beg your pardon, is not affected by temperature. So enzymes, so maybe this becomes zero. So there's, molecular transformations are easier for enzymes when compared, but they're very specific, and they can, catalyze other substrate activities, but very sensitive. You'll have to watch, all my experiments from the past 2 days. They were kind of very sensitive. You see, I added enzyme first, then I added the magnesium sulfate, Epsom salt, and then it split it. So, and here I was thinking that, together all the three would work as it had before. Very sensitive. So that is so delta S may, activation energy is based on um, delta S, an increase in entropy, and that could be, I suppose, considered as a minimal level. So minus T, temperature of the body, that becomes like a really small quantity, but very important, because delta G is a small quantity to begin with. It's something between 3 to 5, that's what Watson says. So I look up Watson, and that will be it for my discussion for today. For luck, because Professor Zanstra back then had found millions of dollars. Have I shown this before? There's for Professor Zanstra. And Justin Trudeau, as Prime Minister, went to Professor Zanstra's lab. Yeah, safety glasses and what have you. Then Justin Trudeau as Prime Minister. Maybe Mark Carney will visit. Now Professor Allen has become a research professor. So delta G values range between 5 and 10 kilocalories per mole, and in vitro experiments could determine all these factors, which would make it easier. These birth defects could be corrected, because otherwise these children are going to be having it hard in life, right? I mean, being different, and children can be cruel too. Children are wonderful creatures, but they can be cruel too. So to each other, the teasing and that sort of thing, and so that might be nice. And anything else? And then that is it, I think. And all right, now part two of this, the shopaholic, sexaholic in me says a quick song. My husband's gone out for a few. Um, we'll just do Spotify. My favorite tunes. Coleman Hawkins.
Signing off. Give a big hand of applause to Golden Hawkins, please. Thank you.